Hello, everyone. Thank you for joining us for a special look at what our restoration teams are doing on a ship in the deep Gulf. My name is Sasha Francis. I'm the Gulf Restoration Education and Outreach Manager from National Marine Sanctuary Foundation, and I will be your host today. Soon we'll get to talk to our friends on a ship named RV Point Sur, which is a 135-foot vessel owned by the University of Southern Mississippi and operated by the Louisiana University's Marine Consortium, or LUMCON. Today, the team is currently located about 110 miles away from the port of Gulfport, their home port. So throughout today's program, please share any questions or comments that come up in the chat, and we'll pause for questions as we go. And we would love to get your feedback on today's program. So we have a short survey you can fill out to tell us a little bit about your experience. So we'll send the link to that in the chat now, and then we'll do it again at the end of the program. We would love to have you fill that out before you log off today so we can help improve these programs. All right, in just a few minutes, we'll connect with folks on board. But first, we're going to talk about where they are and why they're out there. So, as you travel deeper in the ocean, less sunlight is able to penetrate the water. Just past recreational scuba diving limits, you reach the mesophotic zone, meaning middle light. Despite the low levels of sunlight, this zone is home to colorful corals, sponges, fish, and invertebrates. And beyond this twilight zone, there's no sunlight at all, as well as colder temperatures and higher pressure. But there are still vibrant habitats full of life at these depths, hundreds to thousands of feet below the surface. These mesophotic and deep benthic, or seafloor ecosystems, are found worldwide, including in the Gulf, which is actually where all of this footage is from. However, the 2010 Deepwater Horizon oil spill dealt a heavy blow to Gulf communities on land and at sea. 11 people lost their lives, and natural resources, wildlife, and habitats across the northern Gulf were severely impacted. Over 770 square miles of deep sea habitat were impacted by the spill. As a result, collaborative restoration projects are underway to help us understand and restore these seafloor ecosystems found below 160 feet deep. These projects are developing new and improved maps of the seafloor to locate these important coral communities. They're learning more about the species that live in these habitats, the environmental conditions they thrive in, and how they may be connected across the Gulf. They're also determining the best methods to encourage deep sea coral growth and reproduction to help restore impacted coral species and the important habitat they create. They're also reducing the threats that these ecosystems face and connecting the habitats and restoration work to the public and natural resource managers through programs like this. This restoration work, made possible by the Deepwater Horizon Natural Resource Damage Assessment Open Ocean Trustees, is led by the National Oceanic and Atmospheric Administration and the Department of the Interior, plus many partners, including a large number of academic institutions and non-governmental organizations. And we're going to chat with some of those partners today. So to learn more about the oil spill and all of the restoration projects, check out the Gulf Spill Restoration website linked in the chat and video description. All right. Now for the fun part. We're going to connect with folks on board so we can see what's happening right now out at sea and learn a little bit more about the specific objectives of this mission. Hi, how are y'all doing today?
Hey, doing great. Thanks, Sasha.
Can you introduce yourselves to the audience and tell us a little bit about your jobs on this mission?
Sure. Hi, everyone. My name is Sarah Tweed. I'm a trust employee at the Smithsonian Institution's National Museum of Natural History, and my role is a research biologist. That's my official heading there, title there. And I'm here out at sea to work on ARMS, or Autonomous Reef Monitoring Structures, which we'll talk about pretty soon.
Hey everyone, my name is Luke McCartan, and I'm a postdoctoral researcher at Lehigh University in Dr. Santiago Herrera's lab. And on this expedition, I'm helping collect the arms from the seafloor and process them so we can learn all about the life at the bottom.
Awesome. So can you tell us a little bit more about how arms are designed and how they work?
Yeah, happy to. I've been working with these for a little bit more than 10 years now, and they're my favorite thing in the world. Um, ARMS is an acronym which stands for Autonomous Reef Monitoring Structure. They're a sampling device originally invented by NOAA scientists to better survey the cryptic or hidden biodiversity in seafloor habitats. They've gone through a couple of iterations through the years, but the current version of ARMS is made of PVC plastic. And these are actually plastic plates that are stacked in a very particular way. You have a base plate at the bottom, and the stack is composed of alternating layers that are open for flow or with crossbars to create small caves. And these structures mimic the nooks and crannies and complexity of the seafloor habitat to encourage organisms to come and move in. So think of them kind of like prefab housing, and we go and we put the ARMS unit on the seafloor, and organisms on the seafloor move inside and make a home there. It's great because it means that the ocean creatures move in, and you can remove that piece of the seafloor without doing any damage or taking something out that you put in yourself. And you can think of it sort of like an underwater condo. So there's different levels. So different creatures like to live in different layers of the unit, or they like to choose the open layer or the closed layer. So you get a great sampling of the biodiversity on the seafloor. And then most critically, the most important thing is that ARMS are the same size and in the same configuration all throughout the world. The Smithsonian runs an ARMS program, which sounds funny to say, where we source the materials for our colleagues, and anyone who wants to use these, put them out to study the seafloor, are using the exact same devices in the exact same size, and they process them in the same way. So what you get is actually a standardized sample of the diversity that lives on the seafloor. And this importantly allows us to compare between locations or compare the biodiversity in one site through time.
That's very cool. Those are some great visuals of seeing them on the seafloor. It's neat. So why do we need ARMS as a special tool to find out what creatures live down there? What are we missing when we can survey a site with an ROV or other types of sampling methods?
Yeah, so I'll point out a couple of things. First, with an ROV or other conventional, video-based survey techniques in the deep ocean, you're only there for the amount of time you can be on the seafloor, right? So you're there for maybe hours, possibly days, but usually just hours or even minutes. And so what you're getting is really just a snapshot of the organisms that are living in that environment at a certain point in time. However, which is especially important for the mobile critters, with the ARMS, you're putting them down for, years. And so that allows time for the organisms to actually come and settle into these structures. So you're really getting a sample of the community that's really integrated over time, so you can capture more biodiversity. And then the other thing is that the ARMS focus on the smaller animals that inhabit the seafloor that we call like the cryptic fauna. So things, in terms of millimeters or microns in their size. And so you're really not able to see those creatures using, conventional ROV-based video surveys. You're only seeing the larger fish or branching corals or maybe sponges that are standing off of the seafloor. But with the ARMS, you're capturing everything that's growing on the seafloor and in all those little nooks and crannies, which is really where most of the biodiversity is.
Awesome. And I know it's spooky season. Happy October. So I think a lot of people watching might be like, cryptic. Do they mean like Mothman? Like, what do you mean by, from a scientific perspective, what do you mean by cryptic versus what I think most people think of?
Cryptic just simply in this case means hidden. And one general rule of biology is that life likes to live with life. And sometimes life likes to live inside of life or on top of life. So what we also mean are organisms like to live together. And a lot of what we find on the ARMS are, for instance, tiny shrimp that live inside of a sponge or a tunicate. You're seeing a picture right now of a type of echinoderm that's related to a sea star called an ophiuroid. And these are commensals on some of the corals you find in the mesophotic zone here in the Gulf. So creatures like to live together. And when they're very small, you won't necessarily find them unless you're looking inside of other creatures that you collect.
Very cool. Yeah, that seems like there's a lot of that in the deep sea. So, creatures living on other creatures, and it's pretty fascinating. All right, thanks for kind of breaking that down for us. So,
Absolutely.
I'm sure a lot of folks are interested in how these arms are processed now that we've seen kind of what they look like, how they're assembled, and learn about what they're helping us understand. Can you walk us through kind of that process of processing the arms?
Certainly. Firstly, it's already a challenge to get these devices off the seafloor once we've put them down. And as you mentioned, Sasha, we're already operating at depths below, most diving depths, and we're working with remotely operated vehicles. So we needed a mechanism to be able to put a box or a crate over top of the device to capture swimming creatures that live inside it to keep them from swimming away. And we also needed this to be constructed in such a way as an ROV could then lift and manipulate the device and get it back to what you're seeing, an elevator platform to bring back to the ship. A colleague at the Smithsonian, Lee White, actually designed this latching crate that goes over the arms unit. And you're seeing some footage here of an ROV manipulating the arms. So once that latching crate goes on, it gets put on this elevator, which actually Luke helped us with this year in bringing that down from Lehigh University. If you want to speak a little bit about that,
Sure,
you can describe it.
So the elevator is a really cool piece of deep sea technology that's rather DIY, which is fun. It can be put together with hardware that you can source from many different places. But essentially, it's this platform with baskets that you can actually put things from the seafloor onto and then send back up to the surface. So on this expedition, there's this really complicated dance between the remotely operated vehicle and the elevator and the lines that connect them to the ship. And so we have to have everyone all on the same page. So the ship, the remotely operated vehicle, and everyone working on the deck of the ship to get the elevator back up to the surface, like you're seeing here. And this technology allows us to pick up multiple arms in a single ROV dive. And so we can get all three of them back up to the surface so they can be processed immediately in the lab.
Yeah, so what you're seeing here is bringing these back on board. And at this point, the clock is running. So we have sea creatures. We've now brought them out of their habitat, and we'd like to keep them alive to make sure that we can sort them and photograph them and process them. So as soon as they're back on the deck, we turn around and we move the arms into bins of water with bubblers and fresh seawater that was taken from the seafloor using a CTD. And it gets back into the lab, and then everything explodes. There's basically 3 types of processing that happens on every single arms unit. The first step is to take the arms apart, wash the plates in that bin of water, and we sort the plates according to where they were in the stack. So we usually call it floor 1 to floor 9, like a hotel. Once we've done that, we then take that water and we put it through a series of sieves. So we scoop that bin water out, we sieve it through these sieves, and that's capturing all of the swimming and mobile creatures that were washed out of the arms and are in the water of that bin. So that's sort of one major station is separating the motile creatures. And we sieve them into multiple size classes. The large ones will be vouchered and become specimens in the museum and provide DNA that we can use to help identify other creatures later. And then the smaller size fractions, which are too small necessarily to see unless with some visual aid, are usually in the sediments that have been swept into the arms. And those sieve fractions get bulk preserved. When I say bulk, it just gets preserved in alcohol or another preservative, all as one big batch of sediment. Those fractions go back to the museum to have DNA extracted from the entire sample. Very similar to filtering water for environmental DNA. And the kind of big step and big push involves the plates themselves. And eventually, those plates will get scraped using handy household paint scrapers. And all of that material growing on the plates will get put into one blender and blended up. And the entire arms fraction that was growing on the plates will be DNA sequenced to identify the community using the DNA. But before we do that,
Oh, Sarah might have froze for a moment, I'm sure. Oh, you're back. Okay, good. Before we do that is what we heard last, leaving us on the edge of our seat.
Got it. Before we do that,
there we go.
We take a series of high resolution images of every single plate and every single surface of each plate so that we have a permanent record of the community that is growing on the arms. So if you follow me, I can take you over to show you what one of these looks like.
Awesome. Lots of teamwork in that time-lapse video too.
Yeah, absolutely.
Very busy on board when y'all are doing that, huh?
Yes, it is.
All right. We got a little mobile cam action. We're going to go see. Oh, hello again.
I traveled really far.
Yeah, you went very far.
The room.
Oh, it's not huge. All right, what do we got here?
What you're seeing here is a stationary camera setup. And when plates come out of the arm stack, they go into this setup and the plates get photographed on a grid. So every single surface of every single plate has 15 images taken of the surface. And we're able to use image editing software to then take those images and stitch them together to make one giant photo mosaic. And what's really powerful about this approach is that you can produce an image of the plate that's basically one-to-one. And you can zoom in to creatures on the plate and identify what's there. We can calculate the amount of different types of animals that grow on the plates. So this plate had a lot of sponge, or this plate has a lot of coral. And we can also use it to look at things like coral recruitment, for instance, like how many baby corals have settled on this arms and made it a home. So if you look over here, this is actually an image of a plate collected from an arms yesterday. This unit was collected in the northeastern part of the Gulf in an area called Alabama Alps, or that we call together Alabama Alps. And this is actually plate 9 bottom. So this plate would have been at the top of the arm stack and the underside of the plate. So think of it as sort of like a ceiling of your home. These things are growing on the underside of one of those plate surfaces. And I can actually zoom into this image closely here.
I'm glad that's not what the underside of the ceiling in my house looks like.
So what we're seeing here on the edge of this plate are some tunicates.
Ooh.
These are stylid tunicates. And this organism here is called a bryozoan, or a moss animal. And I can take you over to the other edge. We see worms. This here is a type of bivalve, sort of like a clam. This here is an oyster, a very, very small one. And actually what we can see here very close to each other, we have 2 coral recruits.
Yay!
One here. Zoom in a little bit more. So this is a scleractinian coral. And actually, I didn't see it before, so this is the power of documenting everything in high res. There's actually a second coral here.
Did you just notice that now while we're looking at it?
I didn't notice it before.
real time observation. How exciting.
And it's great because, this arms has been preserved for DNA already. So by taking these high resolution images, if we have any questions or we want to quantify different amounts of different organisms, we're going to go back to the photos, since these plates have now been cleaned and are ready to be reassembled into another unit.
Amazing. That's very cool. So these arms are being deployed at many different sites across the Gulf, right? So how does doing that contribute to restoration of these lower light or no light coral habitats that were injured by the oil spill?
So like I mentioned before, because arms are the same size and the same materials, they provide a standardized sample. And the reason that's important is it allows you to make comparisons. So if we put arms out in different locations in the Gulf, we have a standard sample of the entire community we can use to make some inferences about how the ecosystem might be slightly different, or how the microhabitat, the little details of where these arms were placed might be different between these two different sites we're comparing. You not only can compare between different locations, you can also use them to compare between times. So in fact, these arms were put down in 2019 by the Cycle program. So what we have here is a 6-year deployment. And if we had other 6-year deployments of these units, we could start to make some comparisons between growth rates, between the type of taxa, between some sort of major differences in how things grow over that time period. We can also use arms to compare how a community changes through time. So if we put down arms in 2023 at different sites and later put some down in 2026, we'd be able to make some estimates about how that community has changed from 2023 and onward. In this instance, these arms are in areas that were known to be impacted by the spill, and we might be able to compare the communities and how they're recovering at these sites to reference sites in other less impacted areas of the Gulf.
Very cool. So it really provides like a really good big picture of these different habitats, how they might be different, but also how they might be connected, right? And it's really a very cool way to have a great understanding of these areas that we're trying to restore, right? Because we want to know what's down there to be able to restore it. Very cool.
Absolutely.
So I saw some pretty cool little crabs and shrimp in those videos we were watching while you were explaining the processing. What's one of the most interesting critters that's been detected in the Gulf using arms, or that you've come across during processing of these things?
That's like,
you're not supposed to pick favorites, right?
Animal. I have too many animals. There are a couple. In some previous arms from the Gulf, we found these interesting little, they look like worms. They're worm-shaped and very colorful, but they're actually a type of mollusk related to snails, and called aplecopherans, which is a bit of a mouthful. And these things are known to live on and or eat corals. And they're poorly described groups, so if you find some, it's likely that they're new to science. But in this instance, they were brightly colored, purple and yellow. So those were really exciting. And I also think that finding associations is really exciting. And whether that's a shrimp found inside of a sponge, I know from units placed in the Gulf and retrieved in 2021, I opened up a clamshell back at the museum to take some tissue to sample DNA, and I found a tiny crab inside this clam. And it turns out it's a special type of crab that lives in bivalves called a pea crab. And it's also likely new to science. So those are really exciting for me. But I know, Luke, what's your favorite from arms?
Oh, I think everything, honestly. It's just so cool to see all of the small creatures that are living in and among them. And it's really interesting to see the different critters and all the different colors they have, especially across the different sites, because things can change. And so yesterday we found this beautiful shrimp that had stripes of red, stripes of like this highlighter yellow tone, which we were kind of talking about as a group as to why these organisms in the deep are actually colored that way. And so when you really go down and look and you see all of the creatures that call the seafloor home, it allows you to start to answer so many interesting questions, just that, really make you think as like a scientist about why life is the way it is.
Very cool. And I know that Sarah mentioned some tiny crabs, and I just remembered our very famous tiny crab. That I think a lot of people might be familiar with. So we're going to put in the chat the really cool Smithsonian article about that world famous tiny crab. We're going to pop that in the chat too. There's also some other links we might include in the chat about Smithsonian's sample going from a sample from the field into the collections, which Sarah touched on a little bit. So let's see if we have any questions so far in the chat. Folks that are watching, please feel free to type some questions in, and we'll do our best to get to those in our last few minutes that we have here. Okay, so wow, there's like a lot that we've learned today. So thank you for sharing that. And I have to just note, are those people behind you just under trash bags right now? We got like a spooky ghost situation, going on back there. What's going on?
Yeah. They're taking photographs, so I think they're trying to isolate light sources in the lab to get.
They're trying to make a dark room on a bright boat. Yes.
We planned. This cruise was planned pretty strategically, and we have colleagues here who are studying different materials and coral settlements, and they've picked up some tile arrays that are very similar in processing to the arms. So we're taking some pictures on the side here of those settlement tiles, and it's hard to get a good image when there's a lot of different glare from different light sources in the lab.
Yeah. I just noticed that behind you, and I was like, people are going to think that there's a ghost on board. And I think we're going to talk some more about those settlement arrays, the other work that's going on this mission tomorrow. I know y'all are heading back to port, so we'll see what we'll be able to feature tomorrow, but we are going to be talking about those tomorrow as well. So
great.
If folks want to tune in tomorrow, you can see what we have for tomorrow. We'll talk more about that. So I know y'all get to go to sea fairly often, it seems. And I've heard it's been a little bit rough on this mission, and the days are usually long no matter when you're out at sea. But more often than not, it's really enjoyable and a cool opportunity to be at sea. So I wanted to ask y'all, what's your favorite part of working at sea when you get to be out there?
Yeah.
All of it. I love seeing live creatures, and it's super exciting to be collecting all of these tiny live creatures from such a difficult habitat to access and study. So in some ways, it's like bringing the seafloor to you when you pick these up and bring them on the ship. And I think being on the ocean while you do it almost gives you even that much more of a close connection to it. Yeah. So we see all the life in the lab throughout the day, and as the day gets longer and transitions into night and you're working, you step out on the back deck and there's sea creatures and life just off the side of the ship. Last night we were even watching some flying fish and some dolphins feeding on those flying fish, which was pretty exciting. I'd never seen that before. Really cool.
I think for me, I agree with everything, obviously, and just the fact of being out here and being excited and doing the science and collecting the samples is just so exciting. But I would also say that like life at sea is a unique experience for sure. And I started going to sea almost 10 years ago, and I really didn't know what to expect. And what I found is that over time, it really feels like a really human experience because you're a part of a team, and that team's mission is to complete scientific objectives, but also just to make sure you get 3 meals in your stomach every day and you get good sleep, which can be a lot. But I find it most similar to like going out with a camping trip with a bunch of people where, you're thinking about what hike are we doing today? What meal are we going to eat this evening? And I think that that's just like a really unique and really lovely experience. And so I love being out at sea, and it's an experience that I would tell everyone to try if you get the chance.
Yeah, that's a really good metaphor. It is kind of like a big camping trip where you get to know each other well. You really have to work together and look out for each other. And I know that usually the food is much better than I eat when I'm camping. Having some amazing chefs on board and keeping you well fed meals and tons of snacks is super crucial. Awesome. I think we do have one question here. How do you decide how far apart the plates should be? Oh, I think within the arms device itself, but also perhaps when you're deploying them, like location wise.
Yeah, so the configuration of a single unit is standardized, and I believe based on the hardware size. So the spacers that you see in cream color that are keeping plates apart are standard hardware size. The crossbars are the same size as those spacers. And what it makes is a really nice workable sized package, you know, about less than a cubic foot of material to work with. It's been found that plates that are smaller, and these plates are approximately 9 by 9 inches, plates that are smaller are a little bit less easy to compare because things settle out a little bit more randomly on a small space. So it turns out this 9 by 9 space is big enough that you get a really good sampling of the entire community and not just what happened to settle on the plate and take over. And then as far as how the things are spaced apart, we always put arms down at one location in triplicate. So we talked about a little bit about standardization, and this is an extra layer where not only do we have one sample at one location, we actually have 3 replicate samples. And as far apart, we usually put them within a few meters of each other. The goal would be if you're standing at any one arms unit, you should be able to see the other 2. And that is a practical matter where when you put something down in the ocean, it is challenging and difficult and to get it back. And storms could sweep things away. Things might get covered with encrusting organisms. So part of the goal there is to have them close enough that they're sampling the same community, far enough apart that you're getting a little bit of spread in that community, but also visible from each other so that when you find one, you're pretty much hopefully going to find the other 2.
Right. Yeah. And we take locations of everything anytime we put anything down, but it's really helpful to have something that's big enough to find. If you're just using, the visual, or you get to that location and somehow it's moved along with, the Gulf being a hurricane zone, that can happen. So it is really nice too, I imagine, to be, oh, well, there's one here, so there must be 2 others, and be able to move around and find them. And that's another great note too about working together and having, all the multiple objectives on one mission, having different assets on that ship as well. So having the ROV working with in tandem with your team with the arms and being able to, help each other out is pretty amazing. Well, I think that's all the questions that we have today. So I just wanted to say a big thank you to y'all for taking some time to join us today and share your expertise with us. That was super fascinating. And tell whoever's in the trash bag, thanks for that laugh. We'll probably chat with whoever's under there tomorrow, hopefully, or at least talk about their work. So tune in with us tomorrow at our live stream. Wherever you found this live stream, you should be able to see on that page what time we're having our live stream tomorrow. So thanks on board for joining us. We'll let you get back to work as I wrap up here. So
thanks so much.
Yeah. So if you like today's event, we have some more live streams, like I mentioned, coming up, not just tomorrow, but throughout the month. So check out the University of Rhode Island's Inner Space Center YouTube page and save those dates. We'll be pasting that in the chat. Although if you're here on the page now, you should be able to see what's next. And to learn more about these restoration projects, find articles, publications, our story map, hear about updates, etc., you can check out this other link in the chat as well. And we'd love to get your feedback again on today's program. So please fill out the quick survey that's linked in the chat, and there's a QR code to it here as well. And thanks so much for joining us, and we hope to see you next time. And if you know anybody that missed this program, the recording should be up at the same link. So come back, send them our way to check it out.